Good morning, everybody. Welcome back to daily lectures on NEET biology and chemistry, and for the All Indian Medical Sciences, and life sciences for the Council for Scientific and Industrial Research that conducts the national eligibility test. I'm Dr. Bobby. Good morning to all of you. Welcome to my daily lectures. We continue with yesterday's subject of recombinant DNA technology. Functional eukaryotic mRNA does not have introns because they have been removed by the splicing machinery of the eukaryotic cell. We're in chapter 3, right, which is the recombinant DNA technology. Please listen to the subject matter proper. Kindly use them for your subject matters. Do not commercialize any aspects of these lectures. Use them to the best benefit of your exams and your future career. Thank you. And continuing with your subject matter proper. The mRNA has a G gap at the 5' end and usually a string of up to 200 adenine residues, poly A, till at the 3' end. Excuse me. I'm just magnifying this a little so I can read it better. Now, the poly A tail provides the means for separating the mRNA fraction of a tissue from the more abundant ribosomal RNA or RNA and transfer RNA tRNA. Short chains of 15 thymidine residues, oligodeoxythymidilic acid, oligo-DT or DT15, if I read that right, are attached to cellulose beads, and the oligo-DT cellulose beads are packed into a column. Cellulose beads, and the oligo-DT cellulose beads are packed into a column. Total RNA extracted from eukaryotic cells or tissues is passed through the oligo-DT cellulose column, and the poly A tails of the mRNA molecules bind by base pairing to the oligo-DT chains. The tRNA and rRNA molecules, which lack poly A tails, pass through the column. The mRNA is removed, eluted from the column by treatment with a buffer that breaks the AT hydrogen bonds, thereby releasing the base paired mRNAs. Before the mRNA molecules can be cloned into a vector, they must be converted to double-stranded DNA. This synthesis is accomplished by using in succession two different kinds of nucleic acid polymerases. A reverse transcriptase synthesizes the first DNA strand. An E. coli DNA polymerase 1 synthesizes the second. After the mRNA fractions are purified, short unattached sequence of oligo-DT molecules are added to the sample along with enzyme reverse transcriptase and the 4 DNTPs. An oligo-DT molecule base pairs with the adenine residues of the poly A tail of an mRNA and provides an available 3' hydroxyl group to prime the synthesis of the first cDNA strand. Reverse transcriptase, which is encoded by certain RNA viruses, retroviruses, uses an RNA strand as a template while directing deoxyribonucleotides into the growing chain. Thus, when A, G, C, or U nucleotide of the template RNA strand is encountered, the complementary deoxyribonucleotide, that is T, C, G, or A, is incorporated into the growing DNA strand. Unfortunately, full-length first cDNA strands are not always produced by reverse transcriptase in vitro. Incomplete first DNA strands are due to the inability of reverse transcriptase to proceed to the ends of long mRNA templates, frequently pausing of the enzyme during synthesis, and intrastrand base paired configurations, secondary structure impeding synthesis. The second Complementary DNA strands are generated by treating the RNA-DNA heteroduplex molecules with ribonuclease H, RNase H, which nicks the mRNA strands, thereby providing a free 3' hydroxyl group for initiation of DNA synthesis. As synthesis of the second strand progresses from the nicks and the 5' exonuclease activity of DNA polymerase 1 removes any nucleotides that are encountered. The eventual length of the second strand depends on the length of the first DNA strand and the location of the nick in the mRNA molecules relative to the 3' end of the first DNA strand. The synthesis of the second strand is often initiated several nucleotides from the 5' end of the mRNA. However, the obtaining some full-length cDNAs for cloning is equally not a problem because most eukaryotic mRNAs have non-coding leader sequences that range from 40 to 80 nucleotides in length and precede the coding sequence. In other words, a few of the cDNAs will have a complete protein coding sequence and a truncated leader sequence. After the second DNA strand by synthesis is terminated, the ends of the cDNA molecules are blunt-ended and repaired or polished with T4 DNA polymerase, which removes 3' extensions and fills in from 3' recessed ends. Chemically synthesized adapters with extensions for restriction endonuclease recognition sequence are ligated to the ends of the cDNA molecules to facilitate cloning of the cDNAs into a vector. A cDNA library is screened by DNA hybridization to identify clones that carry a specific plasmid cDNA construct. Positive clones must be re-examined further to determine which ones carries the complete coding sequence for the target protein. Once a full-length cDNA is discovered, the sequence can be retrieved and cloned into a vector that is designed to support its expression in a prokaryotic cell. As noted above, the stranded cDNA synthesis protocols produce both complete full-length and incomplete molecules. Unfortunately, much time and effort can be spent on identifying clones of a cDNA library with full-length sequences. Various strategies have been devised to overcome this. This is figure 3.27, which shows the synthesis of cDNA. A oligo-DT primer is added to a purified mRNA preparation, and reverse transcriptase with the 4D and TPs is used for the production of a complementary cDNA strand from the RNA template. Reverse transcriptase does not always produce full-length cDNA copies from every mRNA template due to mRNA secondary structure. That is happen loops or the alifactors. For second strand DNA synthesis, the mRNA is nicked by RNase H, which creates initiation sites for E. coli DNA polymerase I. The 5' exonuclease activity of DNA polymerase I removes both RNA and DNA sequences that are encountered as synthesis from the nick closest to the 5' end of the mRNA progresses. The cDNA molecules are end repaired with T4 DNA polymerase, and adapters containing restriction enzyme recognition sequences are ligated to the ends to increase the efficiency of cloning. So take screenshots of this, and it should help you. This inconvenience, which is taken care of, a method for generating full-length cDNA molecules that is based on a PCR is represented in chapter 4. Here, a multi-step procedure for capturing full-length first-strand cDNAs that are used as templates for the synthesis of second strands is described in figure 3.28. Briefly, the primer for first-strand DNA synthesis is the polydeoxythymidyllic acid, poly-DT, sequence at the 3' end of a synthetic nucleic acid sequence that also contains a recognition site for a restriction endonuclease. This dual function oligonucleotide is called a primer adapter. The disaccharide cellulose is added to the reverse transcriptase reaction to stabilize the enzyme and allow DNA synthesis to proceed at a high temperature. Secondary structure due to intrastrand base pairing in the mRNAs is disrupted by high temperature, and the likelihood that complete molecules will be synthesized is increased. In addition, one of the 4 DNTPs in the reverse transcriptase reaction mixture is 5-methyl-D-CDP, which is incorporated into the growing strand. The presence of methyl groups in one strand hemimethylation of double-stranded DNA protects the DNA from being cleaved by certain restriction endonucleases. This DNA modification is important for the final step of the procedure. At first, the first strand is synthesized. Biotin is chemically attached to the ribose sugars of the cap nucleotide at the 5' end of the nucleotide at the 3' end of the mRNA molecules. Deoxyribose is not biotinylated under these conditions. Next, the hybrid RNA and DNA molecules are treated with RNase 1. This enzyme cleaves single-stranded RNA. It does not attack RNA that is base paired with DNA or DNA strands. As a result, both the 5' single-stranded regions of the mRNA with complete cDNAs and the non-paired segments, the poly A tails, the mRNA molecules are degraded. The mRNA strands of completely synthesized cDNA strands are not affected by this enzyme. The sample is then mixed with the streptavidin-coated magnetic beads. Biotin has a high affinity for streptavidin. After RNase 1 treatment, the only biotinylated RNA-DNA hybrid molecules that remain are those with the biotinylated cap. In other words, only full-length cDNAs are captured because the 5' end of the mRNA is base paired with the cDNA, as therefore protected from cleavage by RNase 1. Leaving the molecule attached, a magnet is used to separate out the beads from solution. Next, the RNA of the streptavidin-bound RNA-DNA hybrids is hydrolyzed with RNase H, which cuts base paired RNA and releases the full-length cDNA strands into solution. Since the sequences at the 3' ends of the first strand cDNAs are not known, a string of guanine nucleotides is added to the 3' hydroxyl ends to provide a complementary sequence for a DNA primer that initiates the synthesis of second cDNA strands. The addition of the guanine nucleotides is performed by the enzyme terminal hydroxynucleotidyltransferase, which adds DNTPs sequentially by a phosphodiester bond formation to the 3' hydroxyl end of a polynucleotide in the absence of a template strand. If only one type of DNTP is present in the terminal transferase reaction mixture, in this case DGDP, a homopolymeric toy tail is formed. After polydeoxyguanilic acid, that's poly-DG, tailing a polydeoxycytidilic acid, poly-DC. Prime adapter is added that base pairs with the poly-DG tail and provides an available 3' hydroxyl group for second strand cDNA synthesis. The adapter portion of this oligonucleotide contains a sequence where another restriction endonuclease site. Second strand cDNA synthesis is carried out at high temperatures with thermostabilized DNA polymerase, RNase H, and DNA ligase, as with the synthesis of the first cDNA strand. Right after the first strand is synthesized, biotin is chemically attached to the ribose sugars of the cap nucleotide at the 5' end and the nucleotide at the 3' end of the mRNA molecules. Deoxyribose is not biotinylated under these conditions. Next, the hybrid RNA DNA molecules are treated with RNase 1. This enzyme decleaves single-stranded RNA. It does not attack RNA that is base paired with DNA or DNA strands. As a result, both the 5' single-stranded regions of the mRNAs with incomplete cDNAs and the non-paired segments of the poly A tails of the mRNA molecules are degraded. The mRNA strands are completely synthesized cDNA strands are not affected by this enzyme. The sample is then mixed with the streptavidin-coated magnetic beads. Biotin has a high affinity for streptavidin after RNase 1 treatment. The only biotinylated RNA-DNA hybrid molecules that remain are those with a biotinylated cap. In other words, only full-length cDNAs are captured because the 5' end of the mRNA is base paired with the cDNA and is therefore protected from cleavage by RNase 1, leaving the biotin molecule attached. A magnet is used to separate out the beads from solution. Next, the RNA of the streptavidin-bound RNA DNA hybrid is hydrolyzed with RNase H, which cuts base paired RNA and releases the full-length cDNA strands into solution. Since the sequences at the 3' ends of the first strand cDNAs are not known, a string of guanine nucleotides is added to the 3' hydroxyl ends to provide a complementary sequence for a DNA primer that initiates the synthesis of second cDNA strands. The addition of the guanine nucleotides is performed by the enzyme terminal deoxynucleotide L-transferase, which adds DNTPs sequentially by a phosphodiester bond formation to the 3' hydroxyl end of a polynucleotide in the absence of a template strand. If only one type of DNTP is present in the terminal transferase reaction mixture, in this case the DGTP, a homopolymeric tail is formed. After polydeoxyguanilic acid, poly-DG tailing, a polydeoxycytidilic acid, poly-DC primer adapter is added to the yeast base that base pairs with the poly-DG tail and provides an available 3' hydroxyl group for second strand cDNA synthesis. The adaptive portion of this oligonucleotide contains a sequence for another restriction nucleoside. Second strand cDNA synthesis is carried out at high temperatures with the thrombostabilized DNA polymerase RNase H and DNA ligase, as with the synthesis of the first DNA strand. This is a figure showing the schematic representation of a method for selecting and cloning full-length cDNA molecules. One is purified mRNA, blue color, is mixed with an oligonucleotide prime adapter with an oligo-DT sequence and restriction endonucleoside, yellow. That is here. Blue with the yellow here. Primer adapter, you have a reverse transcriptase, a complete first strand, and then you have an incomplete first strand. And reverse second will be reverse transcriptase synthesizes the first cDNA strand red with 5-methyl-D-CDP red boxes as one of the floor DNTPs. Both incomplete and complete DNA strands are synthesized. 3 biotin light blue boxes labeled B. All these is attached to the enzyme mRNA molecules. RNA base I susceptible regions are marked by square brackets. 4 single-stranded segments of RNA are degraded by RNase. Biotinylated molecules bind to a streptobiotin-coated magnetic beads. After RNase, when treated with full-length RNA DNA hybrids as biotinylated, and therefore they bind to streptavidin. Incomplete DNAs are not biotinylated and do not bind to streptavidin. 6. The RNase H treatment degrades the RNA of streptavidin-bound RNA DNA hybrids and releases full-length first strand of cDNA molecules. 7 will be a poly-DG tail is added to the 3' hydroxyl tail end of the first cDNA strand. And 8 would be an oligonucleotide primer adapter with no oligo-deoxycytidilic acid oligo-DC sequence and a restriction endonucleoside green pairs with the oligo-deoxyguanilic acid oligo-DG tail and provides a 3' hydroxyl group for synthesis of the second DNA strand by DNA polymerase. During the synthesis of the second DNA strand by DNA polymerase, none of the DNTPs are methylated. RNase H removes by remaining base paired RNA and DNA ligase joins DNA segments that were synthesized internally from bits of mRNA that escaped degradation. The oligo-DC primer adapter sequence that acts as a template for DNA synthesis from the 3' hydroxyl group at the end of the poly-DG tail. And 10. The final full-length cDNA are cut with 2 restriction endonucleases, one for each and cloned into a vector that is complementary extensions. Heavy methylation protects the cDNA from cleavage by the restriction endonucleases that they are used for cloning, because these enzymes cannot cut methylated restriction endonucleosides. And now on to the next page. The high temperature diminishes intrastrand folding and increases the efficiency of synthesis of a full-length strand. The DCTP in the DNTP mixture used for second-strand synthesis, not methylated RNAs. H removes any RNA that escaped the previous treatment, and the DNA ligase joins segments that were primed by remaining bits of RNA. The final product is a full-length double-stranded cDNA that is furnished with non-methylated restriction endonuclease recognition sites at both ends. These sites are cleaved with the appropriate restriction endonucleases and cloned into a vector that has a complementary extension. Hemimethylation generated by incorporation of methylated molecules, nucleotides, during the synthesis of the first cDNA strand protects cDNA from cleavage if it contains the same restriction endonuclease sites that are used for cloning, because the restriction endonucleases do not cut at methylated sites. Vectors for cloning large pieces of DNA, bacteriophage lambda vectors. The plasmid-based vectors used for cloning DNA molecules generally carry up to 10 kb of inserted DNA. However, for the formation of a library, it is often helpful to be able to maintain large pieces of DNA. To this end, various high-capacity cloning systems have been developed. The E. coli virus, bacteriophage or phage alpha lambda, has been engineered to be a vector for inserts in the range of 15 to 20 kb. After bacteriophage lambda infects E. coli by injection of its DNA, two possibilities exist. It can enter a lytic cycle that, after 20 minutes, leads to the lysis of the host cell and the release of about 100 phage particles. Alternatively, the injected bacteriophage lambda DNA can be integrated into the E. coli chromosomes as a prophage and can be maintained more or less indefinitely as a benign guest lysogen through successive cell divisions, figure 3.29. However, under conditions of nutritional or environmental stress, the chromosomally integrated bacteriophage lambda DNA is excised and enters a lytic cycle. The bacteriophage lambda DNA is about 50 kb in length, of which approximately 20 kb is essential for the integration, excision I by E events. For forming genomic libraries, it was reasoned that this 20 kb of DNA could be replaced with the 20 kb of cloned DNA, and subsequently this recombined DNA molecule could be perpetuated as a recombinant bacteriophage lambda through compulsory lytic cycles. To appreciate how bacteriophage lambda cloning systems function, some understanding of the molecular aspects of the lytic cycle is necessary. This is a table of the insert capacities of some commonly used vectors. Vector system is plasmid, host cell E. coli, insert capacity kb 11.1 to 10. And bacteriophage lambda by E. coli or E. coli, that is 10 to 20. Cosmid E. coli 35 to 45. Phosmid E. coli 35 to 45. Bacteriophage B1 E. coli 80 to 100. BHC E. coli 50 to 300. Plasmid bacteriophage derived artificial chromosome E. coli 100 to 300. Yeast artificial chromosome yeast 100 to 2000. Human artificial chromosome cultured human cells greater than 2000. The next page, the life cycle of a bacteriophage lambda. Lysogeny occurs when the bacteriophage lambda genome, red, becomes integrated into the host chromosome. Blue, otherwise, the lytic cycle is initiated, resulting in the production of and release by cell lysis of about 100 bacteriophage particles about 20 minutes after infection. Infective bacteriophage lambda consists of a tubular protein tail with a protein tail fiber and a protein head packed with 50 kb of DNA. The production and assembly of the heads and tails and the packaging of DNA are a highly coordinated sequence of events. The DNA within the head of a lambda particle to a 50 kb linear molecule with a 12 base single-stranded extension at the 5' end of each strand. These extensions are called cohesive ends because they contain sequences that are complementary to each other. After the injection of the lambda DNA through the tail into E. coli, the cos ends base pair to form a circular DNA molecule. During the early phase of the lytic cycle, DNA replication from the circular molecule creates a linear form of lambda DNA, and that is composed of several contiguous lengths of 50 kb units that concatenamers 50, let's have figure 3.3a. Each newly assembled head is filled with 1 kb unit of DNA, and finally the tail assembly is added to complete the formation of an infective particle. As figure 3.3, the volume of the bacteriophage lambda head is sufficient for about 50 kb if less than 38 kb of DNA is packed into a head. A non-infective bacteriophage particle is produced. More than 52 kb of DNA cannot fit into a head. The location of the cos sequences, which are 50 kb apart, and the multiple length linear alpha DNA and the lambda DNA ensures that each head receives the correct amount of DNA. Located at the opening of the head is an enzyme that recognizes a double-stranded cos sequence and cuts the DNA at the site as the DNA is inserted into the head. By mixing purified empty heads, bacteriophage lambda DNA 50 kb and tail assemblies, infective particles are produced in a reaction tube. One of the many bacteriophage lambda cloning vectors that have been devised has two BAM-HI sites and that flank the IE region. When purified DNA from this bacteriophage is cut with BAM-HI, 3 segments are created. The left arm L region contains the genetic information for the production of heads and tails. The right arm R region carries the genes for DNA replication and the cell lysis. And the middle IE fragment that has the genes for the integration and excision processes. The objective of this genetic engineering is on to the next page. Just before we finish that sentence, let me finish on this figure. Packaging of bacteriophage lambda DNA into heads during the lytic cycle A. DNA replication from the circular form of bacteriophage lambda creates a linear form that has contiguous multiple lengths concatenamers of bacteriophage DNA with units of approximately 50 kb each. Each newly assembled head is filled with a 50 kb unit of lambda DNA before the tail assembly is attached. And now the objective of this genetic engineering protocol is to replace the middle IE segment of the lambda DNA with clone DNA that is approximately 20 kb in length, figure 3.31. The BAM-HI treated bacteriophage lambda DNA sample is enriched for L and R arms by size fractionation and removal of IE segments. The source DNA is cut with either BAM-HI or SAW3AI or A1, and DNA pieces that are 15 to 20 kb in length are isolated. The digested source DNA and the LNR regions are combined and incubated with T4 DNA ligase. The empty bacteriophage heads and tail assemblies are added. Under these conditions, 50 kb units of DNA with insert DNA flanked by the L and R regions with cosins are packaged into the heads, and infective bacteriophage particles are formed. Other products from the ligation reaction cannot be packaged because they are too large greater than 52 kb, or too small, less than 38 kb. Also, any 50 kb DNA molecules without a functional region of a replication and cousins cannot be perpetuated. Recombinant bacteriophage lambda undergoes lytic cycles and is maintained by growth in E. coli. To identify the recombinant bacteriophage that carries the target gene in the bacteriophage lambda library, the individual zone of lysis blocks each continue next page to finish the figure here. A bacteriophage lambda cloning system. Bacteriophage lambda is engineered to have two BAM-HI sites that flank the IE region. For cloning, the source DNA is cut with the BAM-HI and fractionated by size to isolate pieces that are about 15 to 20 kb long. The bacteriophage lambda DNA is also cut with BAM-HI, and size fractionation removes the IE segment. The L&R arms plus the 15 to 20 kb source DNA molecules are mixed with T4 DNA ligase. The ligation reaction produces a number of different DNA molecules, including ligated DNA, source DNA only, combined L&R arms only, and molecules that have a source DNA molecule flanked by L&R arms. So this is the fractionate, the retain and discard, right, leading to the T4 DNA ligase. The last molecules are packaged into bacteriophage heads in vitro, and infective particles are formed after the addition of tail assemblies. The recombined bacteriophage lambda is perpetuated by infection of E. coli. Some 50 kb source DNA ligation products may be packaged into heads, but since this DNA lacks both the functional region of replication and cousins, it cannot be perpetuated. Other ligation products are either too small or too large to be packaged. For some bacteriophage lambda cloning systems not shown here, high packaging efficiency is achieved by setting the conditions of ligation to favor concatemer formation to imitate how the phage heads are normally filled. Each of which contains recombinant bacteriophage are lifted onto a matrix and screened with either DNA probes or antibodies. For DNA hybridization, the bacteriophage proteins are removed and the DNA is denatured and bound to a matrix. For immunological assays, the proteins encoded by clone genes that are synthesized during the lytic cycle, along with bacteriophage and bacterial proteins, are transferred with a plaque and subsequently bound to the matrix. On the basis of the sites in the matrix that give positive responses, corresponding plaques the original plate subculture to provide a source of selected recombinant bacteriophage that's individually cultured in E. coli. Plasmids. Cloning vectors called plasmids can carry about 45 kb of cloned DNA and are maintained as plasmids in E. coli. Plasmids combine the properties of plasmids and bacteriophage lambda vectors. For example, the commonly used plasmid PLFR5 has two cousins from bacteriophage lambda, flanking a SEA1 restriction endonuclease site, a multiple cloning site with 6 unique recognition sites, HIND3, PSD1, SAL1, BMHI1, BAMH1, SMA1, and EcoRI, an origin of DNA replications, and a TET gene, as seen on figure 3.32. Pieces of DNA that are approximately 45 kb in length are purified by sucrose density gradient centrifugation from a partial BAMH1 digestion of the source DNA. The PLFR5 DNA, which is the 6 kb kilobyte base pair, is cleaved initially with SCA1 and then the BAMH1. The final two DNA samples are mixed and ligated. Some of the ligated products have an 45 kb DNA piece inserted between the two fragments that are derived from the digestions of the PLFR5 DNA. That is clone LFR5 DNA. These molecules are about 50 kb long and have cos sequences that are about 50 kb apart. Consequently, these DNA constructs are successfully packaged into bacteriophage alpha lambda heads in vitro. Reconstituted PLFR5 with our inserted DNA is not packaged After the assembly of bacteriophage particles, the DNA is delivered by infection to E. coli. Once inside the host cell, the ACOS ends, which were cleaved during the in vitro packaging base pair and enable the linear DNA to circularize. This circular form is stable, and the cloned DNA is maintained as a plasmid insert DNA construct because the vector DNA contains a complete set of plasmid functions. Moreover, the TET gene allows colonies that carry the plasmid to grow in the presence of tetracycline. Non-transformed cells are sensitive to tetracycline and die. A plasmid is a kind of plasmid vector that carries up to 40 kilobytes of insert KB, that's essentially base pairs, of insert DNA. A plasmid is a kind of plasmid vector that carries up to 40 kb of insert DNA and a COS site for in vitro bacteriophage lambda packaging. The difference between a plasmid and a plasmid is that the original replication of a plasmid is derived from the E. coli F factor, cytoplasmid, hence the name. The advantage of plasmids is that they are very stable single copy vectors, whereas plasmids are maintained at higher copy numbers, which often leads to deletions or rearrangements of parts of the insert DNA. Other E. coli bacteriophages have been used for creating vectors. For example, the genome of the PL bacteriophage is 115 kb long. The PL-B1 vector system can carry 80 to 100 kb of inserted DNA. The advantages of using plasmids and other vectors derived from bacteriophages are twofold. First, because the capacity of these vectors is greater than that of plasmids, gene clusters and large genes can be cloned. Second, a large insert in the cloning vehicle means that fewer clones of a genomic library have to be screened for a specific gene. This is the next figure. Please take screenshots and make notes. This is figure 3.32, a plasmid cloning system. The plasmid contains an E. coli region of replication, ORI, that's ORI, that allows the plasmid to be maintained as a plasmid in E. coli, two intact COS sites closely flanking a unique SEA1 site, a unique BAMH1 site near but outside one of the COS sites, and a TET gene. The source DNA is cut with BAMH1 and fractionated by size to isolate molecules that are about 45 kilobase pairs long. The plasmid DNA is cut with SEA1 and BAMH1. The two DNA sites are mixed and treated with T4 DNA ligase. After ligation, some of the joined DNA molecules will have a 45 kilobyte piece of DNA inserted into the BAMH1 site of the plasmid. When this happens, the two COS sequences are about 50 kb apart. These molecules are packaged into bacteriophage lambda heads in vitro, and infective particles are formed after the addition of tail assemblies. Infective bacteriophage lambda delivers a linearized DNA molecule with COS extensions in E. coli. After entry into the host cell, the COS ends base pair, and the DNA ligase of the host cell seals the niche. The circular DNA molecule that is created in this way persists as a plasmid in the host cell. In this case, transformed cells can be identified because they are resistant to the antibiotic tetracycline. This is table 3.8, which shows the estimated numbers of clones required for 99% probability of representation of every DNA region in a genomic library for various organisms and cloning vectors. Organism, genome size BP, clone BR322 of 5 kb, number of clones bacteriophage, lambda 70 kb, plasmid 35 kb, and BAC is 150 kb. So it's short for E. coli, yeast, fruit fly, rice, humans, and frogs, right? I just mentioned the first and the last but one, that is E. coli is 4.6 and 10 raised to 8 genomic size, and the clone BR322 is of 5 kb type, kilobase pairs is about 4234. And the number of clones here for bacteriophages, 1243, plasmid is 602, and BAC is 138. If you go to humans, it's 3.3 into 10 raised to 9 genomic size. And the clone BR322 at 5 kb is 3,090,475 base pairs. And the bacteriophage lambda is 898,392, plasmid is 434,053, and BACs are 101,258. For the cloning vectors, the sizes in parentheses are the average sizes of the insert DNA. Now, the high-capacity bacterial vector systems. A vector system that carries very large inserts greater than 100 kilobase pairs is helpful for the analysis of complex eukaryotic genomes. For example, these types of vector systems are indispensable for creating libraries, for genome sequencing, and for carrying one or more intact genes on a single insert. In contrast to a small insert library, a large insert genomic library is more likely to include all of the genetic material of the organism with fewer clones to maintain. Table 3.8. It shows that a low copy number E. coli plasmid vector that is based on the P1 bacteriophage cloning system has been devised for cloning DNA molecules that are from 100 to 300 kb in length. The DNA insert vector constructs of this system are called bacteriophage P1-derived artificial chromosomes. Similarly, the F plasmid, F factor replicon, sex plasmid or fertility plasmid of E. coli, which is present at 1 or 2 copies per cell, has been used along with the LAGZ selection system of the PUC vector to construct an extremely stable cloning vector that carries DNA inserts from 50 to 300 kb in length. The F plasmid-based DNA insert vector constructs, which are used extensively, are called YACs. Genetic transformation in prokaryotes. Transferring DNA into E. coli. Transformation is the process of introducing free DNA into a bacterial cell. And for E. coli, which is the main host cell for recombinant DNA research, the uptake of plasmid DNA is usually achieved by treating mid-log phase cells with ice-cold calcium chloride, that is CaCl2, and then exposing them for 2 minutes to a high temperature of 42 degrees centigrade. This treatment creates transient openings in the cell wall that enable DNA molecules to enter the cytoplasm. This method has maximum transformation frequency of about 1 transformed cell per 1,000 cells, that is 10 raised to minus 3. The transformation efficiency is approximately 10 raised to 7 to 10 raised to 8 transform colonies per microgram of intact plasmid DNA. Although 100% transformation frequency will be ideal, selection schemes that enable plasmid transformation cells to be readily identified overcome the drawback of a low transformation frequency. In some of the bacteria, competence occurs naturally and in some cases can be enhanced by the use of specific growth media or growth conditions. These bacteria are usually easily transformed. Other DNA delivery systems must be used for bacteria that are either refractory to chemically induced competence or are not naturally competent.